Auzu billahi minash shaitani rajim. Bismillahir Rahmanir Rahim. Sallallahu alayhi wasallam. We are discussing our topic of enzyme and book resources for this topic is Harper 31st edition, page 156, chapter 7, 8, and Lippincott 7th edition, page 53, chapter number 5, and the Essentials of Medical Biochemistry, volume 1, page 139, and chapter number 6. Today, inshallah, we will discuss the enzyme substrate complex. In fact, we will discuss the chemistry of enzyme and substrate complex in a series of lecture. Inshallah, we will sum up this discussion in a 3 consecutive lectures. First of all, we will discuss how the enzyme and substrate interact with each another or hold each another. In fact, there are 2 models for this interaction, which is lock and key model and induced fit model. In the second lecture, we will discuss that how after interaction of enzyme and substrate, the energy of activation decreases. There is a famous graph for this mechanism of action of enzyme, and we will inshallah discuss that graph in that lecture. We will also discuss the free energy, energy of activation, and different dynamics of that particular graph. In the third and last lecture of this series, enzyme substrate complex, we will discuss through which mechanism, in fact 4 principles or mechanisms, the enzyme decreases the energy of activation and increases the rate of reaction. Okay, let's start with our first lecture of this series, which is explanation of lock and key model and induced fit model. Okay, lock and key or Fisher model is self-explanatory, and old model, it explains how a specific enzyme engages a specific substrate and convert this substrate into product. In this model, the lock is formed by the cleft, which we explained in our previous lecture, that is the active site basically, by a cleft pocket on the surface of enzyme called active site. It provides this active site in fact provides the environment wherein a chemical transformation take place. Basically, in this model, the enzyme behave as we suppose that enzyme behave as a lock and substrate as a key. There is a conformational complementary site for the substrates within enzyme, and after the lock and key complex, means enzyme and substrate complex or ES complex, a conformational changes occurs within that site and enzyme is transferred into and substrate is converted into product. We have discussed in a previous lecture the properties of this active site. As we discussed earlier, the enzymes are protein structure, and this protein structure of enzyme scaffold to support and position the active site, because enzymes, basically the enzymes are bigger product of protein and it have only a small portion of active site. This happens because the active site contains a particular sequence for or complementary sequence for its substrates, and this sequence or these amino acids are dispersed throughout the enzymes. Enzyme have to opt a three-dimensional or particular position to scaffold or to make the active site in which in which amino acids are present in a particular sequence. Like this one, this is the proteomic structure of lactic dehydrogenase LDH, and the whole protein structure of LDH scaffold or arrange itself in a three-dimensional structure to arrange to make an active site for their substrates lactate and NADH, and this active site make a complementary structure of amino acids in active sites. So the whole enzyme scaffold its position to make an active site for the substrate lock and key model. We have just explained that the whole protein structure of enzyme make a scaffold position to arrange amino acids in its active sites and engages the substrate in a lock and key model or lock and key fashion. That's an old model for engagement or interaction of substrate and enzyme complex. A new model with advanced theories later on explained because the limitation of this lock and key model. It has merits that within the active site substrates are brought into close proximity where the cofactors present in that enzymes or prosthetic present in that enzyme engages with that substrate and perform their function. This interaction, lock and key interaction, generates an environment where polarity, hydrophobicity, acidity, alkalinity can differ from the surrounding cytoplasm. In fact, this interaction creates a complete new microenvironment within that cytoplasmic or cellular environment, where there may be a complete change of polarity or hydrophobicity may occur, and substrate may then changes into product. But there are limitations of this model, that this model failed to account for the dynamic changes that accompany the catalytic transformations occurs within that active site due to its rigidity or complementary structure of lock and key model. That in this case the enzyme failed to explain the conformational changes that we will discuss in a later lecture that happens during the mechanism of action. To address these limitations of lock and key model, later on Daniel Koshland introduced a new model of substrate and enzyme interaction called induced fit model. In induced fit model states that as substrates bind to an enzyme, they induces a conformational change that is analogous to placing a hand into glove. in which we can assume glove as enzyme and substrate as hand. Like we can assume enzyme as a glove in which the substrates fit in as a hand, and it provides a conformational or induction of conformational changes at active sites. And after that, suppose in this diagram, this is the substrates, and here the enzyme and substrate fit in induced fit model, and substrate is converted into the product. After the interaction, there is induction of conformational change within the enzyme, but specificity of the enzyme does not change. The right hand glove will fit into the right hand, it will not fit into the left hand. The fingers will fit into the finger of the that gloves. So the only thing which explained further is flexibility of this complex of substrate and enzyme in this model, which was the limitation of lock and key model. When the active site on the enzyme makes contact with proper substrate, the enzyme molds itself to shape of the molecule. Suppose A and B are substrates, and their product are, that is AB is the substrate and their products are A and B. Enzyme is lyase in this example. So what happens that when the substrates AB interact with the enzyme, it molds up and break the AB into A and B and change the substrate into product A and B. So we conclude our today's topic that lock and key model. In a local key model, there are complementary sites of substrates on enzyme active sites, but in induced fit model it provides a flexibility, and after the interaction of enzyme with the substrate, there is induction of there is induced conformational changes in the enzyme, but in both cases, after the completion of reaction, the enzyme is unchanged. Again, the today's take home message is stay home, stay safe, and clean your hand repeatedly. Goodbye.